Welcome back to the Back Bay. And now we're really pleased to be able to introduce a panel on AAV delivery, opportunities and issues. The panel is going to be co-moderated by Dr. Xandra Breakefield, who's a geneticist. She's appointed in the MGH Department of Neurology, and she's a past president of the American Society for Cell and Gene Therapy. And her fellow moderator is Dr. Florian Eichler, a neurologist also appointed in the Department of Neurology at MGH, and he is the director for the Center for Rare Neurological Diseases at MGH. Xandra and Florian, take it away.
Hi, I'm Xandra Breakefield. I'm a professor of neurology at Mass General Hospital and Harvard Medical School, and I'd really like to welcome our distinguished panelists to discuss this exciting topic with AAV kind of leading the front in gene therapy.
I'm Florian Eichler. I'm delighted to be here with Xandra. I'm director of the Center for Rare Neurological Diseases at MGH, and I'm going to briefly introduce this panel. The topic is that of AAV delivery, really highlighting what we've seen over the past 10 years as a sea change for our patients in terms of availability, access, and therapies coming to the fore based on AAV modalities. Together with that, a number of challenges arise, and this panel is really meant to collectively address opportunities and challenges and how we can innovate, collaborate together. Xandra?
Well, maybe the panel can introduce themselves first. Can you go ahead?
Jennifer?
Hi, everyone. I'm Jen Farmer. I am the Chief Executive Officer at the Friedreich's Ataxia Research Alliance.
Manny?
Hi, everybody. I'm Manny Simons. I'm the CEO and co-founder of Akuos.
And Matt?
And I'm Matt Fletcher, Senior Vice President, Division Head, Gene Therapy Research and Technical Operations at Astellas Gene Therapies.
Great. Thanks, Xandra, for keeping me on track.
So we have some people that are right on the front lines of bringing these vectors into therapy in patients. And I guess my first question would be, what kind of challenges are you facing in this, in these clinical trials and getting them set up? Jen, you want to start?
Sure, thank you. Friedreich's ataxia is a multi-system disease, and so as we think about AAV approaches to a disease like Friedreich's ataxia, we need to target multiple organ systems: the brain, the spinal cord, the heart, the muscle, and so in developing these programs, one of the challenges we've run up against is the biodistribution. Being able to, where we are right now, consider a one-time therapy, how do you get to all of those organ systems with a one-time therapy? And, there are some approaches with delivery that can help solve some of these challenges, but it really is balance between delivery and dose and safety, when we think about even multiple or dual routes of administration to try and reach all of these targets. And so, at least in a disease like FA, biodistribution, I see as one of the major challenges while we try and advance these therapies.
Yeah, that was kind of the first time I thought about doing, as a first injection, doing it at multiple sites at the same time, which is, I think, a very interesting concept. And Manny, how about you?
Sure, yeah, so just for any who aren't familiar, Akuos is focused on AAV gene therapy for inner ear conditions, and everything that we work on at Akuos is a locally administered AAV gene therapy delivered to the fluid of the inner ear. So this is considered a novel route of administration because there aren't any drugs, biologics, or fluids that are approved for administration directly to the inner ear. And so as we were thinking about developing the first gene therapies for inner ear conditions. Delivery is a critical early question to address, and I think has been one of the reasons there hasn't been more done in this space up until now. So despite similarities between the eye and the ear, the inner ear is an enclosed compartment, there's reduced immune surveillance, the target cells are non-dividing. So all the things that are well suited for AAV gene therapy, but it's really lagged in large part because there hasn't been a translatable way to deliver vector distributed throughout the full length of the cochlea and the inner ear and in a translationally relevant way. And as we think about going from an initial proof of concept study in a mouse to what's going to be, how do we choose the right dose in the human population? How do we administer vector in a way that is going to be accessible to surgeons, not just in early clinical development, but throughout the pivotal process of development and into a setting where it's broadly accessible to as many potential patients who could benefit. This is it's where a lot of work within the company has had to focus. There's not infrastructure today throughout contract research organizations to do that translational work in the inner ear. And so I think there's some adjacency to this theme to what Jen was just mentioning, in that delivery is critical for gene therapies, whether it's finding the appropriate ways to access multiple organs, or even within one focal organ like the inner ear, making sure you're doing it in a way with the right animal models throughout the development process, so that what you're bringing to humans ultimately is something that has a potential to deliver really meaningful benefit.
Yeah, I mean, it's an interesting thought too. Eventually, if it did work, you'd probably want to go into both ears at once, right? So
that's right.
Yeah.
Yep, that's exactly right. So thinking about that from early in development, I think is important.
Yep. And Matt, how about you?
Yeah, thanks. I think I want to pick up on something Jen touched on around the biodistribution, and generally the idea that where we are right now in gene therapy, when you look at the trials going on, the vast majority of them are using first generation AAVs that come with them certain limitations. The fact that, the AAVs that power most of our gene therapies are the ones that came out of the very early work by people like Jim Wilson and Luc Vandenberg, identifying naturally occurring variants. And the reality of the situation, by just utilizing what we've been able to find in nature, is we haven't perfectly optimized that system, which leads to potential deficits in our ability to reach our target tissue, our ability to do that at a reasonable dose range. And so I think, in fact, as we move into the next generation, if we can better engineer our capsids and better engineer our viruses to solve transduction and biodistribution, it's going to go a long way in moving the field forward. It's going to address not just issues of efficacy, the fact that we can actually get our target, our transgene to the right target, to the right tissue, that we can get it and transduce enough cells to make a difference. But ultimately, if we're doing that at a more efficient rate, it means that our doses are lower, which then get a lower viral exposure, potentially seeing benefits in safety. And then ultimately, lower doses also mean a lower pressure on manufacturing capacity, which is a major bottleneck right now, as well as cost of goods, which then goes into the price and accessibility of the gene therapies. So I think just in continuing to optimize our delivery tool here and be able to drive it to the next level is going to make a big difference across the board in a number of these areas.
Yeah, I think that the concept of lowering the dose is a really important one, especially since very high doses can be very toxic. Well, I'll let Florian have a go here.
Yeah, no, it's exciting to hear about all this progress in terms of potency and ability of new vectors to be more efficient and achieve the kind of biodistribution that Matt was talking about, and particularly for the compartments that Manny needs to address as well. I was curious, as you are thinking about different diseases that you're going to be tackling, how you are addressing the interaction with the disease biology, in particular the different stages of disease that you will be facing, which in the human setting may differ quite a bit from the mouse. And so how are you thinking of this, particularly in terms of biomarkers that might guide efficacy and safety? And I think this is an opportunity to think about collaborations that you might need or be looking for to help solve future problems. So maybe again, I'll start with Jen.
Thanks, Florian. I think your point about collaboration is a key one. That you know, the issue of developing biomarkers based on the biology of the disease, you know, are not specific to any one program. And I think, you know, this is the way in which we can bring multi-stakeholders together in a pre-competitive way to, you know, try and tackle some of these problems so that we can get early reads on efficacy in gene therapy approaches. And through Thera, we've put together some of these kind of multi-stakeholder collaborations around biomarkers. For example, we brought a lot of our academic researchers together with several of our industry partners to think about, you know, how do we develop better neuroimaging biomarkers in FA, and, you know, put together a pre-competitive study that was looking at brain and spinal cord imaging and multimodalities, because these, you know, in some of these rare diseases, we can't do multiple studies, and we need to share this kind of information across programs. And so where we can come together and collaborate to solve some of these problems to improve the clinical development process across programs, I think is really important.
Yeah, I think you're right. This is also the expectation of the patient population, that you want to, you know, be able to collaborate and learn from each other as you move different programs forward. I'm as a neurologist, I'm always thinking about, you know, is the underlying disease going to be unmasked by side effects, and specifically with the DRG toxicity that you're seeing with some of the AAV gene therapy. I think that's a concern. So I think it's great to collaborate and move together with that. Manny, what are your thoughts here?
Yeah, something that is really important to us and guided some of the early decisions around genes of interest, indications to focus on that we thought would be well suited to AAV gene therapy approaches, are trying to identify where there is biology and assessment measures that are conserved across species. And in just looking at for hearing specifically, the monogenic hearing disorders, so hearing loss associated with mutations in individual genes, there are very good, highly representative mouse models that have the same phenotype as what you see in the human population, same pathophysiology, similar natural history progression. Mice tend to not be as reliable to things that are more complex etiology, like things associated with age or accumulation of mitochondrial deficits, things like that, where they probably represent a large population of human inner ear or hearing deficit, and probably just less well represented in something like a mouse compared to the monogenic hearing disorders. So that affects how we think about the mix of different non-clinical or preclinical models that we use. So when mice are an appropriate model, they can be tremendously valuable in helping advance a potential therapy to and through clinical trials. In some cases, they need to be augmented by human genetic data, or even human data from similar biologics that aren't necessarily gene therapies, but help kind of clarify or validate the mechanism. For us, in hearing, there's an auditory brainstem response, or ABR, which is it's a non-invasive objective measure of auditory function. So it's average neural activity from the synapse of cochlear nerve fibers in the cochlea to projecting to the brain. And they're used clinically, they're relied upon by audiologists, otologists. It's included in newborn hearing screening. It's used regularly in a clinical setting. And we can use that assessment in mouse studies. We can use it in non-human primate toxicology studies. And that's a really clear way for us to evaluate throughout the development process One, it can be an indication of tolerability throughout across species, but also indications of recovery of meaningful auditory function. So having those types of tools is really valuable. What we don't yet have in the hearing space is something that can identify when there are still viable target cells, even when the cochlea isn't functioning properly. So your auditory brainstem response may be absent, but the cells underlying that function could still be there, viable, just not behaving the way they should. So that could be a really attractive opportunity for a gene therapy if you can restore function without having to rebuild architecture. And so as we think about new tools that could be really valuable in this space, being able to measure or assess the viability of underlying cells, even when their functional output is missing. So that's something that we're continuing to work on.
That's great. Oh, and lovely to hear of this physiology, Manny. That's very elegant. I'm slightly jealous, thinking if I had the same for DRG neurons, wouldn't that be a blessing? But and I think as we try to translate, from mouse to humans in future, we're going to have more trials, right? And hopefully there'll be a life cycle of learning from one trial to the next and shared experience. So I think that will hopefully also go a long way. Matt, what are your thoughts here?
No, thanks, Florian. I think, as transformative as we think and hope gene therapy can and will be, there are just some aspects of good drug development, successful drug development, that doesn't change regardless of the modality. And I think that really is true when you talk about biomarkers of efficacy and safety, that ultimately we need really robust measures of these associated with our programs to both be able to successfully move through early development, but then into the clinic and be able to demonstrate success. And, the problem of having these kind of tools available are just exacerbated when you work in the rare disease space, right? This is we don't have the benefit of potentially decades worth of clinical trials that have validated numerous endpoints and really, develop new imaging technologies focused on those particular diseases that in many times, you're working in a space with diseases that are poorly characterized and poorly understood. And that's where I think it does emphasize that need for collaboration and working together, between not just companies, but also with the patient groups, which often are the holders of most of this knowledge and science, as well as academics and across biotech and pharma. I think one really good example of where this has happened is in Duchenne's muscular dystrophy. There's been a couple different pre-competitive consortiums that have come together to solve potential issues in this area. One such group came together to focus on sharing methodologies for measuring and quantitating dystrophin, which again, this is a key part of most of the gene therapies, as well as even some of the small molecule and large molecule programs. But yet we continue to struggle as an industry on how to actually determine whether or not we're doing the thing that we hope to be doing, increasing dystrophin. And so the groups came together, shared best practices, and have really been able to push that sort of approach forward. Similarly, in another competitive consortium, groups have come together to share their clinical trial data, particularly the placebo-controlled data, which helps both in understanding placebo effect within those populations, being able to control for it, but also getting more robust natural history data that powers the entire industry and everyone working together. So I think in a number of ways, that sets a nice template that many of us could try to follow and continue to work in this area.
Mm-hmm. And I think the common theme here is understanding thresholds of health and disease as relating to exactly the amount of gene correction we require to turn the tide of organ function. So I think it's great to see you reflecting on both the platform technology side as well as the disease biology here. I'll turn back to Xandra for any additional questions.
Yeah, I was just wondering how important, and any of you could answer this, how important do you think it is to have a large animal model of your disease state to kind of really see how well you're getting distribution, et cetera? I don't know, anyone was fine.
I could start just briefly. And I think what you said, Xander, in phrasing the question is the key part, is the biodistribution. I think one thing that we have seen over and over again is that rodents are poor predictors of biodistribution for AAV when you move into non-human primates and in humans. And so therefore, I think the rodent models still have an important place in the preclinical process, particularly answering questions of disease mechanism and correction. But these sorts of questions around biodistribution and dose, I think generally we've seen that they've been inadequate. And so then it does leave us really dependent on non-human primate studies to get these key questions answered. And that's where, to the heart of your question, I think we do need to continue to explore other types of large animal models to be able to see whether or not there are other tools out there besides non-human primates. I think generally there's an interest across the field to reduce use of non-human primates in our studies, but also just as you look at what's happened because of COVID, there's been a supply issue as well, which has held back a lot of our science and a lot of our drug discovery. So we need it from a practical standpoint as well as ethical. So I think continued research into other animal models like mini pigs could be quite useful long-term for us in enabling the field.
I agree with that, and I think that the distinction you made between whether your model is you're using it to demonstrate a prospect for benefit, or like the ability to address the disorder, or for distribution, tolerability, delivery to the right cells in appropriate amounts that help inform dosing, I think is important, and it probably is condition or target gene specific, right? So for us, when we have high confidence that a mouse model can answer the prospect for benefit question or the biology question, we still rely heavily on larger animals, specifically non-human primates, because for the reasons you mentioned, we don't think the mouse is going to give us the answers we need on those. So you need the non-human primate for distribution, tropism, conformation, help inform dose selection for human studies. I think that's important.
Yeah, maybe to bridge from the non-human primate and large animal studies into questions around immunogenicity, because often in those studies, we're not, at least in non-human primates, employing steroids or other immune modulators in the same way. How do you think of progress in the coming years as linked to understanding of immunogenicity? And I'm particularly interested in Jen's thoughts also in terms of the patient population and their willingness to take on certain, risks in weighing it to certain benefits. I always think about Friedreich's ataxia as a disease where there are different organs that are going to be targeted. And are you going to see redosing occur? And is that going to bring questions around immune responses? How do you think of that in the field?
I think that is another challenge that we certainly still have, and even a good number of the treatable population are also adults, right? So they're already going to have a good number of the patient population will already have pre-existing antibodies. So to even be eligible for first therapy, they are likely going to need some sort of immunosuppression, or we're going to have to work around this immunogenicity issue in some way. I think if this can be managed with immunosuppression medications and approaches, I think, the patient community, it weighs that against what the potential benefit is long-term, with a single therapy. And if we can get to the target tissues and, have that significant treatment effect, then I think you can get to a place where, you can take on a little bit more risk with the immunosuppression for the potential benefit of the therapy. But, again, it's a lot of back and forth between managing different benefits for different risks. It is quite complicated.
And I think for a lot of these diseases, you want to go into children, and then the tissues are going to keep growing, and they're going to lose the AAV, so you're going to have to go back in. So it's definitely a challenge for the future.
I do think, going back to something, Matt, you mentioned at the outset, I think even if you're within the capsids, even if you cannot engineer out of a capsid like a immune reactivity, whether it's neutralizing antibodies or some other immune response, being able to achieve a given level of benefit at the lowest dose possible, I think is going to be important. And I think in the earliest stages of the field, it's kind of as much vector as you need to get the desired effect. And if there are opportunities to significantly reduce how much vector can achieve the desired protein expression, that I think will have really meaningful benefits from just immune system perspective.
I guess for all of you, like, how far are you from clinical trials at this point, do you think?
In the Friedreich's ataxia community, we've got several programs that are in preclinical development that are in IND enabling studies. And so we're anticipating being in clinical trials next year, early next year, for several programs. So, and there's, a lot of excitement around that and a lot to learn.
We at AAV, we are we're completing our IND enabling studies for the lead program. This is for a form of genetic hearing loss called otoferlin mediated hearing loss, and expect to file the IND first half of next year.
That's great. Wow.
And then obviously for us, we were in the clinic with a couple programs. Obviously our most advanced is the X-linked myotubular myopathy program, as well as we're also dosing patients now for Pompe disease as well.
That's great. I think we're kind of winding up here. Any last words or words of encouragement or caution or?
I would say it's so exciting to see the options emerge for many of these rare diseases. I just heard of that, I've never been able to see these kinds of modalities come to the fore. So I just want to congratulate everybody to having pushed the field forward. So excited to see your progress in the coming years.
Yeah, and I mean, thanks to all of you. That was really an exciting discussion, and as Florian said, very encouraging for so many of these diseases where there was no hope at all.
Yeah, thank you. Thanks for having us.
Thanks very much.